internships for the summer in the so-called USM Beer Lab. And check them out. They're paid internships. It's actually a really good experience. That's Katie Knox. She's a pre-med student, yet she worked in a brewery. So there, okay? And then next, we go to this. So we're going to focus on this today. I'll hand back the quizzes at the end of class. And today we'll do our first concept chat. So we'll get into that in about an hour, where we just team up with a partner and talk about what we learned that was important. We talked a little bit about chapter 1 and and we'll talk more about it. And in chapter 1, there's a section that introduces some cell biology, and I introduced that a little bit, where we talked a little bit about centrifuges and separating the nucleus from other cell parts. It's a big part of cell biology. What we're going to talk about today is really the basis of those videos, and it's molecular biology. So molecular biology is about molecules, and by far the favorite molecule is DNA. The field of molecular biology was born in the 1940s and 50s when Watson and Crick discovered the structure of DNA. There were already a few molecular biologists in the 1940s that had preceded their discovery of the structure of DNA in the 1950s. And those early molecular biologists were establishing experimental evidence that DNA was the primary heritable molecule. So Watson and Crick got the structure, and even just that later that same decade, scientists were working on this genetic regulation, and they were working on it in bacteria. That's why chapter 7 starts with bacteria and transcription regulation, because a long time ago, some really early molecular biologists were working that out. Like that, okay? So here's some stuff about molecular biology. It's physical, always. It's car mechanics, always. And most of our courses, molecular biology, it's cause and effect relationships, physical relationships like that. Like this protein binds to that protein all the time. That's all it ever is. And the next thing is, it's a kind of reductionism. And reductionism means the whole is not greater than the sum of its parts. And that's a pretty bold statement, right? When you say, I'm a car mechanic of life, and human beings are nothing more than a bunch of fancy tractor parts. That's a pretty bold statement. The reason it's true is a lot of people miss out on why it's true. They're like, no, the whole is often greater than the sum of its parts, right? Complicated systems are more complicated than the sum of its parts. In molecular biology, here's the key thing, is we're not saying the whole is the list of its parts, it's the sum of its parts. And molecular biologists sum up the parts in a variety of ways. Here's one way. They start out by looking at simpler systems. So they understand the parts in a human by looking at bacteria and understanding those parts. Here's another way they do it, we've already talked about, that's true the only thing we can talk about is evolution. And the sum of its parts are always to understand the relationship to these parts in the history of evolution and the history of life on Earth. So when it's summing up the parts, there's a lot of things to talk about. It's literally just the parts. Back to the original word, molecular biology, the parts are molecules, and molecular biologists want to understand those entirely, down at the atomic level. Not just like, oh, it looks like a screwdriver, it must be functioning like a screwdriver. What does it look like in detail? So it's a pretty intense and pretty dogmatic. The earlier molecular biologists were super mean. Super mean. Like there was a guy named Max, and if he came in and the seats were all taken, he would just point to somebody and say, get out, you won't understand this, and take their seat. Yeah, Watson and Crick were really, they sound nice, but they're really, really aggressive people, like that, where this is stupid, like that, that's just stupid, like that. Kind of like some really gruff car mechanics where they're just like, no, that's not going to work, You're going to need a new engine. Yeah, you're not going to drive down the highway any longer in this junk car, right? Okay, so the whole is not greater than the sum of its parts. Don't worry about it, okay? Then the next thing is they really lean on what is called a model organism. And that is, if you want to understand humans, we have other creatures we can understand first and best. And that's called the model organism approach, and that's dominant in biology today. Scientists study simple organisms to understand more complicated organisms. They study a simple organism, and then they spread out into some other organisms, knowing what they learned in that first organism. And how can they do that? And the answer is evolution. Evolution is the guarantor of the model organism approach. it's guaranteed that we'll understand their body by looking at a mouse because of our evolutionary relatedness with them. So that's the next thing. And the second section in chapter 1 is about model organisms. It lists some of those and some features of them and stuff like that. And that's why it's there. It's a little introduction to molecular biology by just focusing on that one feature of molecular biology, which is they go after these simple model systems like that. Yep. Okay. So then the next thing is the research is done in labs. So they bring it into a controlled environment and it gets really taken down to a real specific controlled setting to take and get one answer at a time, little tiny answer. The original molecular biologists were trained as physicists, and their attitude was biology is just physics and chemistry. And we're just going to do experiments in the lab by breaking things down to the minimum system level and understanding them like tiny, tiny little bricks at a time. Then the last thing, and this is kind of a surprising feature of molecular biology, and that is that over the years, scientists have made biological discoveries just about how nature works and just out, and they're in the real world. And they've discovered something, and either they or someone else has very quickly said, we could take this discovery and use it as a tool, as a general tool to help us make more discoveries. And that's really, really common in molecular biology, and it's one of the key things that's really driven it forward, because nature is full of really awesome, interesting things that can be valuable when turned into an experimental tool. And here's an example of that. So in the clay model videos, I say, this is a mouse, and it's a mouse that's had modified gene, DNA added to it. The DNA gets inserted, gets incorporated into the chromosome. So this is called a mouse strain. It will forever reproduce babies that have these same modified genes. The mouse is called a transgenic animal, and it's because it has genes in it that are modified genes that were built in a test tube that are called transgenes. So we put some transgenes into an animal or plant, and it's called a transgenic organism. When you go to the grocery store and you look at GMO, genetically modified organisms, food, they're transgenic plants and animals. Yep, so that's what that is. And so we want to talk about that today. And in talking about it, the gene that gets put in is part of a gene from this jellyfish that lives in Puget Sound by Seattle. And a scientist, a marine biologist, studied that back in the 1960s and 70s. And he was just trying to figure out why it glowed. Why did this little jellyfish glow in the dark? Yep. And I lived in Seattle before I came here. My daughters were really little there. We'd go to the beach. We'd catch these. They're called aquaria. They're about that big. We take them back to our apartment. In the bathroom, there were no windows. We'd close the door, and we'd look at them glowing in there. And then, as a dad, I had to drive all the freaking way back to the ocean. I said freaking, back to the ocean to put them back. Yeah, it's like across Seattle is almost impossible to drive, but I diligently do that. And so that's they are. And this scientist was just curious, and he figured it out. And what he figured out is that there's a gene, and the gene product is a protein that glows. So another scientist really realized that he could take the jellyfish gene, and he could put it into another organism. So we could modify the jellyfish gene, make a transgene in a test tube, and then put it into another host organism, make a transgenic animal. That transgenic animal would have all its normal genes, perfectly healthy, just had one additional gene, and it was this transgene, and the transgene included the jellyfish part that when made into a protein, the protein would glow. So it's really important to appreciate that in this mouse embryo, this transgenic embryo, during its embryonic and fetal development, the cells start to glow. And the thing we're going to focus on is the muscles start to glow red. What's going on with these different colors is another scientist, a third scientist, took the original gene from jellyfish and mutated the gene so that the protein products would glow different colors. The original gene coded for a protein that glowed a kind of green color. It was called green fluorescent protein, GFP. That scientist just looked for mutants in the original jellyfish gene and looked for different colors. So we got different colors out of it. So this transgenic mouse has 3 different transgenes in it. that glow different colors that mark, I would say, the muscles, the tendons, maybe the nerves, like that. So expressed in different cells. In the early stage, that's a transgenic animal, the same strain, and none of it's glowing. It has the genes, but the genes do not glow, only the protein products glow. And so only the red is showing up when muscles start to form. Because what the scientists did in a test tube is they took the transcription unit from the jellyfish that would code for the protein that will glow, and then they joined it in a test tube to the DNA from the regulatory region of another mouse gene. And the mouse gene they picked, they wanted to have the gene expressed in the muscle as the muscle formed, and then it would glow. So that would show them when muscles start to form. So does anybody have a suggestion of what would be a good regulatory region of a gene from a mouse to add to the transcription unit of the jellyfish gene? What gene would you use?
Like the myogenin gene?
Yeah, so that was our quiz. Our quiz was to set it up so you understand that myogenin gene is made of two parts. The transcription unit that codes for the myogenin protein, and the regulatory region that tells where, when, and how much of that gene to produce. And all we have to do is take the DNA for the myogenin regulatory region and join it to the jellyfish transcription unit, and it will now be activated. So the first drawing we're going to do is we're going to build that gene on paper to show you that. So if you haven't got paper and markers already, get some different colored markers, because the first drawing we're going to do is just build the first trans gene. Okay? And I'll do it up here, and it's really simple, but the idea is to emphasize this physical molecular biology, this reductionism, this focus on the cause and effect relationships. And the way to do it is to start out, and you could use, if you want to use 2 or 3 pieces of paper, you can, because it helps spread it out and separate it. And I'll just do it on one piece up here, but what I'm going to do is I'm going to say, we have to pick a color, to pick a color, and I'm going to make one color, and this is going to be the jellyfish gene. And see, I'm drawing in these boxes, and I'm not going to bother to label them anymore. It's just a way of seeing that that's the region, that's the transcription unit, and if it's a coding gene, it's going to have a coding region in it. Then there's the promoter right next to the transcription unit, and the enhancer, and these are the cis regulatory region and the transcription unit, 2 parts of any gene. So always draw it like that, but you need to be able to draw it and label it and explain all those parts on a test. But we had a quiz on that, so you're solid on that, but just be aware of that's what that is. And this is that jellyfish gene. So it's DNA from a jellyfish, and we purified it, and it's the jellyfish GFP gene. When it gets transcribed in the jellyfish into RNA, that RNA is mRNA because it's a coding gene. So it's going to code for messenger RNA, and that messenger RNA is going to be used by the ribosome in the cytoplasm to translate the messenger RNA into a protein, and the protein is, I guess we'll make it green again just to show you that it's the protein, and it's the GFP protein, and we make a bunch of it. And it glows, just automatically fluoresces on its own. That's just the nature of it, that's why they picked it. So again, the DNA, the RNA, they do not glow, but the protein product glows. So we're just going to take just the gene, so just the DNA itself is going to be what we get. We get that, we purify it, and we were just talking about DNA purification, so we do it that way and talked about it before class. And then we got to purify this gene, and this is going to be the mouse myogenin gene. And we picked the mouse because that's where we're going to put it in. We want to have the regulatory work in a mouse, so this is mouse myogenin. And we want that myogenin because its regulatory region is what we want to regulate our transgene. So these are 2 genes. They're made of DNA. That's just DNA, long DNA. The DNA is probably about 30,000 nucleotides long, so we've zoomed way out, and we're looking at 2 genes, and they fit our definition. And so what we're gonna do is, in the test tube, is we're going to cut this part of this gene and move it up here to a new test tube. And so that's that jellyfish gene transcription unit, and it's DNA, purified DNA. And then we're going to cut this region out of the myogenin gene from mouse, and we're going to join those together. And a key concept is that the structure, biochemical structure of DNA is the same in every organism on our planet. So we can make a hybrid gene with two different species. It works just fine. And so that's the mouse myogenin gene. And we're going to put that into a mouse. And we're just going to add it into the mouse's chromosomes. And so it's going to be part of the mouse's chromosome. And that's going to make our transgenic animal. It's an extra gene. The mouse still has its normal wild type myogenin gene. Actually, one from mom, one from dad. It just has this new gene. And so then just draw this in for our last part of this, and that is, here's MyoD protein. And the MyoD protein is happy to bind to this and activate it. And when it activates it, the jellyfish gene gets expressed, and we start to make this GFP. Now remember, in the videos it's red because it was a mutant allele of the GFP that glowed red, RFP, but it just starts to glow. So this is a transgene, right? So we call that a transgene, and the mouse organism is a transgenic mouse. And there are lots of different reasons, lots of different purposes for making a transgene. This is just one. And people would like to call this transgene, they like to call it a reporter gene. And they like to call it a reporter because it's reporting something. And think of the things it's reporting. It's reporting when muscle differentiation starts, right? So you can use it to study muscles. Before the cells glow, no muscles. They're myoblasts or other cells. Then it starts to go, muscle differentiation has begun. We're not going to hear the heartbeat until the animal starts to glow because the heartbeat is a muscle. So we got to make the muscle first. Yep. So that's one reporter. Here's another reporter thing it's doing. It's helping us understand the MyoD protein. Remember, when MyoD gene gets expressed, it kind of immediately goes to work. And this is its job. So we would be able to detect MyoD protein in the glowing cells, but nowhere else. Every other cell in the organism has the gene and has always had the gene, but is not making MyoD, and consequently the cells are not glowing. Right? Then we're also using it to study myogenin, because the myogenin gene is activated. If we looked at the regular myogenin genes in that same transgenic mice, we would see, MyoD is binding there too. When a transcription factor is made, there are thousands and thousands of molecules of it made, and it can bind to all the target enhancers in the nucleus and just light them all up, get them going. So at exactly the same moment that the myogenin gene is activated, the transgene is activated. Another way to think about it is, again, you have two copies of myogenin in your cells, one from mom, one from dad. And they always get expressed at the same time because there's lots of MyoD, and it binds to mom's myogenin and dad's myogenin, and it can bind to that transgene, and it can bind to more transgenes because it's plenty of MyoD protein to bind anywhere. So that's our first drawing. It's our first attempt at showing you that. And you see how they're taking advantage of a discovery. And one of the discoveries is that GFP. So the scientist who studied was actually a guy from Japan. That he studied, he was a marine biologist. He's like, I'm curious, why does this little jellyfish glow? And a scientist in New York City read the paper and said, oh my gosh, if that's how that jellyfish glows, if it's just a protein that's glowing, I could put that into my little nematode worm I'm studying and could get it to glow. Yeah. And both those scientists won a Nobel Prize because it's just like, that is a cool tool. So discoveries get turned into tools. There's more discovered here too. One is the universal nature of DNA allows us to put DNA into any species because it's the same molecule. Another is every gene is composed of two parts. So we can splice genes together and make these hybrid genes. They could be two different species, mouse and jellyfish. They could be two different, functional parts of a gene, and they work together. Hi, Alex.
You said that the mouse myogenin gene is the reporter gene, right? That's
Nope, this is the reporter gene. The
The
transgene is the reporter gene.
Okay.
Yep, the mouse myogenin gene is just the normal mouse gene. We built a transgene, and we can call it a reporter because it's reporting by making a glowing product, reporting that it's making muscle, reporting that MyoD is active, reporting that myogenin is getting turned on. And so whatever the scientist is studying in those categories, it's a powerful tool to further that analysis. Okay? People have questions about that. That's our first drawing. Okay? We're going to do kind of 3 separate drawings today. Okay? So here comes the stuff that we're going to talk about. So there's the jellyfish. There's a transgenic zebrafish fish. Transcription unit from a jellyfish. And the DNA doesn't glow. It's the protein that glows, and that protein is normally called GFP. Yep. Okay. Okay. So now we're going to make now we're going to start over and make a second example of it. And it's going to be the same kind of example. It's going to be a cell that starts to glow because we're going to put the jellyfish gene into it. Okay? Before we start that drawing, I'm going to just show you this, and that is that, remember the FOXO-protein. So we talked about this first day of class, and it's in chapter 17, so we haven't really read about it too much yet. But it's a transcription factor. So it means it's like MyoD and myogenin. We also learned about ELK. We also started talking a little bit about the estrogen receptor. Those are all transcription factors, and we're making a list of those. They're always proteins. And FOXO has this feature. A transcription factor needs to get into the nucleus to function because it binds to DNA and regulates gene transcription. Transcription factor's function is to regulate transcription. And here we see that FOXO can get into the nucleus and do its job under these conditions. However, you have an external growth factor outside the cell and bind to a receptor, and through this pathway, FOXO gets phosphorylated and it gets sequestered, it gets trapped in the cytoplasm so it can't do its job. So we talked about how the protein is in a different subcellular location. In the absence of the growth factor, the FOXO is in the nucleus. In the presence of the growth factor, the FOXO is out of the nucleus. Okay? So we just drew a transgene, and when that transgene makes the GFP protein in muscles, the GFP protein floats throughout the cell. So the entire muscle glows green or red. Here, we're going to make a transgene using FOXO that will glow wherever FOXO is located. Okay? So it doesn't just make the whole cell glow. It's not just floating around in the cell. It's actually showing us where FOXO is. Okay? So we're going to make that one. It's very much the same drawing, but we just want to make a separate one because we want to help bring home these concepts, Okay? So here, we're just going to do the same style thing where we start out with jellyfish GFP over here. So that's the same gene we start with, just wild type normal jellyfish GFP gene. And we purify that from the jellyfish and we get that DNA in a test tube. So it's a gene, physical. And then over here, this time, we're going to have FOXO. And we're going to be working in mouse. We're going to make a transgenic mouse, so we'll work with mouse FOXO gene. And here, what we're going to do is we're going to take the whole entire FOXO gene in the test tube. So up here is where we're going to build the transgene, and it's going to be the entire FOXO gene. We just moved it up here. And what we're going to do in this case is we're going to go to the jellyfish GFP and we're going to find the transcription unit, and within the transcription unit is the coding region that has the codons for the individual amino acids that will be the protein. And so what we're gonna do is we're gonna take that entire coding region and we're gonna cut it out with some molecular scissors, just that whole thing. And what we're gonna do is we're going to insert that piece up here, right into the FOXO gene. And what we need to do is just need to now add the coding region for FOXO. So that's the coding region that codes for the FOXO protein. And what we're gonna do is we're gonna insert that new piece of GFP right near the end of the FOXO coding region. So we're just gonna add this together and we're gonna make it like that. So we make two coding regions in one. I already mentioned that DNA is universal in life. The DNA genetic code, the different codons, they're almost universal in all of life. So when this gets put in, this trans gene gets put into a mouse, we make a transgenic mouse strain that will forever, with all its offspring, have this transgene in it. And once again, it's a normal FOXO gene, and it's just an additional one. Here's the normal FOXO gene, the mouse is fine, this is just extra gene. And when it gets expressed, wherever FOXO is expressed, what happens is it gets transcribed into RNA, and then that mRNA gets translated into a protein. Oh, and the protein, so there's the ribosome translating this into a protein, and the protein looks like this. The protein is FOXO / GFP. And so because of the universal nature of the genetic code, we can make any foreign protein, and we can make that protein connected to the host transgenic mouse's own protein. And so that's another example of a different kind of reporter. And this one shows us where is the FOXO protein located. In the cell. Because anytime FOXO gets made, the FOXO hybrid transgene will get made, and the FOXO protein will glow. And wherever it's located will glow, okay? So that's just that drawing, pretty simple, but it's a really significant add-on because now we want to look at this. So we talked earlier that you can use a Downs homogenizer, and the Downs homogenizer has a plunger that fits into the homogenizing tube, and the plunger is such that the distance between the bulb of the plunger and the wall of the tube is too small for the cells to fit. And so instead of passing by, the cells are broken open. But the nucleus stays intact. So we just take some mouse cells out that are glowing, and then we just grind them in this homogenizer. That breaks the cell, but leaves the nucleus intact, and then we just centrifuge it. So this are pellets of the nucleus. This is everything else from the cell, the extract, okay? So if we did that, under what conditions would the pellet glow?
If it was inside the nucleus?
Yeah, if it's inside the nucleus, and when is FOXO inside the nucleus?
When it's activated.
When it's
Not phosphorylated.
not phosphorylated. So if in the absence of growth factor, FOXO goes into the nucleus. Then it would be here if there was a growth factor present, right? So what's this reporting? It's reporting the location of FOXO and the presence of a growth factor, right? We could study the growth factor by studying where the glow is because it's a response to the presence of the growth factor. Yeah, you, Sarah?
So is it just showing us where the FOXO gene is when there is not a growth factor?
Yeah, either way, it's showing us that switch. And you said where the FOXO gene is. And this is the tricky thing that I mentioned before, where on our answers, we have to be really careful to use the right words. because it's not the FOXO gene, it's the FOXO protein, and it's the transgenic FOXO protein. That's the thing that's glowing. This animal has the regular FOXO protein, and that's going in or out of the nucleus, depending on the growth factor, but that's not reporting anything. The reporting is the glow, just a natural glow. And you just look at this, and you go like, that's glowing, right? So in this, it's going up here, it was in the cytoplasm, the growth factor is present. If it's going down here, it's in the nucleus, the growth factor is absent. So yeah, right on. And there's a question up here.
I was wondering if, so the transcription unit, they add the GFP to that. Did they take anything out of the mouse FOXO gene, like out of its transcription unit? Like, is there a certain amount of space, like you have to take X amount of base
Nope.
out to put
No.
in?
And somebody might ask, when you make these kind of hybrid proteins, do they work? And the answer is sometimes they do, sometimes they don't. Sometimes the FOXO protein is messed up by having this big GFP protein hanging off of it. But it's surprisingly often, they just work. If they don't work, then you can start fiddling it. You could put the GFP at the other end of the protein, or in the middle. Yep. Yeah, but they don't need to remove any part of either proteins. Yep. Yep. Okay, so that's our second drawing assignment. Both of them are building transgenes, but you see the difference? The first one, it's just the whole cell that's glowing, because the first one is just GFP, and it's just floating around. So the whole muscle cell glows. The second one, we've actually built it into existing protein, and the glow will be wherever that protein is. The FOXO protein is really dragging the glow with it, wherever it goes. Yep? Yeah. And so scientists do that, okay? And we're gonna make a third drawing, but we're gonna talk about some stuff before we get to it. The thing I wanna say here is that that's great, and on Thursday, one of your classmates, Sam, is waving. Sam is actually gonna tell us about building another kind of transgene like the FOXO GFP transgene. And Sam is going to talk to us 4 times, and I just talked to him a year ago about doing this because he's really interested in these kind of topics. So he happens to be in this class, but I've had other students over the years explain it. And so he's just going to tell us in 4 meetings about a certain kind of cancer therapy. And the cancer therapy is called CAR T-cell therapy. It's used to treat cancers, and it actually appears as a very short figure in this section in our chapter 20. So chapter 20 that we'll get to, I always forget the numbering because the numbering shifted from 8 to 9. It might be chapter 19. It's the last chapter in our book. And that's where it talks about different cancer therapies. I think the chapter is called Cancer. And this is a really good example because to get this done, on Thursday, Sam's going to talk to the start of class just briefly that they built a transgene. And the transgene story is one like the FOXO GFP. They're going to stick together coding regions to make a special transgenic protein. It's not a reporter. They're not using it with GFP. They're not trying to make it glow. They're trying to make a super protein. So they make this super protein DNA, and then the next time he visits, he's going to tell us that the super protein, the transgene, makes a special receptor. And that's in chapter 17. So we'll hear from Sam, and it's a specific example from chapter 17. And then chapters 18 and 19, I think it's 16, 17, 18, 19, 17, 18, are how cells respond to receptors, and this is what that CAR T cell therapy is they take a transgene, they put it into human cells, and they get the super protein to help that human cell fight cancer. And so this will be the cell response to that that receptor. The cell response for FOXO is it either gets to go into the nucleus or not. So cell responses vary. And then the fourth one is just kind of like a clinical story about, well, what is happening with this work? And the whole point of that is to provide an example of a person who is like our like example. The more you can spread your learning around, the better. So Sam's going to provide this example that's an overarching example of this cancer therapy that's a nice story to tell in a series of visits, okay? So that's coming up, and it's awesome, and thank you, Sam. And you know, it's nothing special. The whole point of it is that Sam is that person. And you know, you'll be able to hang your learning on that example, and we'll go through it. And you can see it builds up, right? So by the final exam, you'll be able to spit it all out. The first step is just appreciating. You can build transgenes, and you know, reporter, that's all for experiments in a lab. You can build them to make super proteins that could fight cancer. Yep, okay, cool, all right. So there's that. So the next thing is we're going to talk about, back to, I think Mallory asked about the estrogen receptor. And so we're going to make our third drawing now, and we're going to start out by drawing the myogenin story, and then we're going to shift over to another transcription factor called the estrogen receptor. It's just abbreviated ER. We talked a little bit about that. It binds the hormone estrogen, and it's involved in sex differentiation of all vertebrates, all female vertebrates, so fish and frogs and dogs and cats and humans. They all differentiate into a female and exposed to estrogen. AR stands for the androgen receptor. Androgen is another word for testosterone. So in all vertebrates, we get females or males based on this hormone. This is a hormone receptor, and this hormone receptor happens to be a transcription factor. So we're going to use that, and we're going to make our third drawing, and we're going to lace that into the drawing. And in this drawing, we're not going to be building transgenes. We're just going to be looking at, first, a drawing of what we learned about in the muscle differentiation videos. And I'm going to sit down to draw this just because it's going to be a drawing. And you're going to build this on a piece of paper, so get some fresh paper. And we're going to have, like, this is going to be a drawing where this is, it's one cell over time. So there's the nuclear membrane. Oh man, maybe this won't be as successful as I hope. So that's the nuclear membrane. And then there must be a cell membrane out here. So there's the cell membrane like that. And that makes this the cytoplasm. And then this is the inside of the nucleus. So we're in the nucleus here. And we make the nucleus big because it's going to have some genes in it. And what we're going to do is we're going to draw 3 different genes. And so they're just going to have that same, gene-looking drawing, right? It's got a transcription unit, there's a little promoter, a little enhancer, like that. Just a little, simplified drawing of a gene. Here's a second gene. Meow, meow, meow. And here's a third gene. I think the colors on this writing pad are cool. That's the only reason I use it. All right, and these are the genes. So this one's going to be MyoD. This one is going to be myogenin. And the third one is a muscle gene. And we could write muscle genes. And those include all the proteins that are in our muscle. And so you've probably taken a course in physiology where you learned about muscles, and the muscle contracts by having muscle fibers slide past each other, and they're polymers of actin sliding past each other. So actin's a protein, there must be an actin gene. That would be an example of muscle gene. And then the little molecular motor that uses ATP is myosin. Myosin's a protein, there must be a myosin gene. And there's lots of other proteins, tropomyosin, troponin T, titin, and so forth, in that muscle fiber. And so all those genes have to get turned on. And so this is a little timeline. So it's really the same cell over time. And you might want to kind of represent that by like maybe a little clock, right? So here's a little clock, that's the first time, and then here's the second time, and here's the third time, like that. It's a time thing, right? And they're just going to go boom, boom, boom. And this is going to be the example of the dominoes falling. So the first thing is that something binds to this. A transcription factor binds to the MyoD gene and activates it. It wasn't activated earlier in pregnancy, earlier in the embryo. Now it's going to get activated. And we said that step is kind of complicated. We don't need to learn about it. We're going to learn about 3 in a row, and we're just going to jump into it and say, now the MyoD gene is activated. And when the MyoD gene gets activated, the transcription unit gets transcribed into RNA, and that RNA leaves the nucleus as mRNA, messenger RNA, and there it is, it's the MyoD messenger RNA, and this is our first time period, and it's going to bind to a ribosome, and it's going to get translated into MyoD protein. And the MyoD protein, there's a lot of it. It gets translated a bunch, and so there's lots and lots, there's plenty of molecules of the MyoD protein at time 1. Okay, and then we've said that pretty much immediately the MyoD protein just goes back into the nucleus, and its job is to bind to myogenin. So it binds to the enhancer of the myogenin gene and it activates it. There are almost for sure other genes that are also activated at the same time. We're just learning about one. But there's plenty of MyoD to bind as many genes as are the targets and activate them all. And so now myogenin is going to get activated. Woo-wee! And so we just pick a color for it. And I don't know what it could be, but it's going to get transcribed into mRNA. So this is the second gene and the sort of second time point, right? Because now a little bit later, the myogenin comes out. And that's myogenin mRNA. And it's going to get grabbed by a ribosome, and it's going to get translated into lots of molecules of myogenin protein. So there's the myogenin protein, yay. We're partying, and after we finish this drawing, we're going to have nap time. So, kindergarten matters. So this myogenin protein we mentioned, well, that's a transcription factor. Its job is in the nucleus. It almost immediately goes back into the nucleus and binds and activates these muscle genes. And those get transcribed into mRNA that goes out of the nucleus, and that's muscle protein mRNA, muscle gene, like could be actin and so forth, like that. Myosin, a bunch of different genes get activated because there's plenty of myogenin molecules to activate everything. And that gets translated by a ribosome into muscle protein. Now we're starting to make muscles, muscle fibers. Pretty soon the cells start to twitch because the muscle fibers are forming and start to contract. So we really have a functional product there. All right? And so you see it just goes boom, boom, boom. All right, so now what we're going to do is you could either redraw this same drawing, or we could just write over it, because it's the exact same drawing, but we're just going to shift the focus. And so we're actually, we should just start over. We're going to start over because we're actually going to just talk about two genes, but it's the same thing. And before we shift over, what we're going to talk about is that this just goes boom, 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 and that's a great first introduction. What we want to talk about next is that there are transcription factors that don't get to do their job right away. So MyoD gets to do its job right away. Think of a transcription factor that doesn't. So FOXO doesn't. If it doesn't get into the nucleus, there's a big pause there. So instead of having FOXO and its target go boom, 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 there's a delay. And that idea of the transcription factor being regulated and creating a delay is probably like a top 10 central concept on our planet for all of life. Because we inherited the DNA we need from our parents for our whole life. And that's true for everything that's alive on our planet, from single-celled bacteria to multicellular plants, animals, and fungus. And we use it over time. So there must be some built-in delays into this system. If you look at the system of the inherited DNA, it includes genes that are going to get activated by the products of other genes. So the DNA has a kind of it's almost like a computer-like program where it's gonna play out over time. And the big story of the time is the delay in activating the transcription factor. And a really epic example of that is the estrogen receptor and the testosterone receptor, because they're made our whole life. But the hormone doesn't show up until we reach puberty. So in that case, the estrogen receptor gene is activated, and it's activated years before it does its job. Like that. Remember we talked about how in male vertebrates, a male vertebrate could get shots of estrogen. The male vertebrate has the estrogen receptor, and it can activate those target genes and get some secondary female sex characteristics. So it's delayed in females through childhood until puberty. It's delayed in males all the time, unless they get exposed to estrogen. So we're going to make that. We're going to make a new drawing of that, and it's just going to have 2 genes, but it's going to be another timeline, okay? Super duper. So really the same layout, where we're going to have a cell membrane on the very far outside, and so that's the cell membrane, and then that must be the cytoplasm, and then we're going to have the nuclear membrane, and that must be the nucleus down at the bottom. And in this example, we're going to use just 2 genes, and the first one is going to be another time one. So off on the left, I'm going to start out with the estrogen receptor gene. So there's the estrogen receptor gene. And there must be some transcription factors that bind to it, and they actually bind to it very early in our development. And they activate the estrogen receptor gene, and so that means it gets transcribed into RNA, and that RNA goes out into the cytoplasm, and that's estrogen mRNA, or we could call it ER mRNA. And we could do an experiment to detect that RNA. So anytime a cell is making estrogen receptor mRNA, we can detect it in an experiment. It's going to bind to the ribosome, and the ribosome is going to translate it into estrogen receptor protein. And we get lots of molecules of it. And the estrogen receptor protein is a transcription factor. So it wants to go back into the nucleus. There's always some estrogen receptor in the cytoplasm, and consequently, some people kind of struggle with understanding what estrogen receptor is doing, because there's always some in the cytoplasm. But its job is in the nucleus, and some of it comes into the nucleus, and then it actually immediately binds to target genes. And there are many target genes of the estrogen receptor. So here's this, and we're going to call these estrogen, for E for estrogen target genes. I think it'd be fair to also call these female puberty genes. These are the genes that get activated for a female girl or a female vertebrate to grow up. And so the estrogen receptor comes in here and binds to it. And because it's binding here, it actually, in the absence of the hormone, and let's just put the hormone up here so we know it's going to come some year. It's not here yet, but that just red box, that's going to be the estrogen hormone. And it's not present yet because it's very young. It's fetus or juvenile. So it's not there yet. In the absence of this hormone, we mentioned before that this is a repressor. It is repressing transcription of these target genes. So in a female juvenile or a male at any time, the estrogen receptor, in the absence of hormone, is repressing the genes. Then the estrogen hormone arrives and it binds to that receptor right there. And you can see the receptor is in the nucleus. So the estrogen receptor is in a gene family. And we remember those are groups of paralogs that emerged through evolutionary time by random gene duplication. And this gene family is called the nuclear receptor gene family. The nuclear receptor gene family, and the estrogen receptor is an example of that. It's bound to estrogen hormone, and that protein is the receptor, the estrogen receptor. And nuclear receptors in humans, that gene family has 48 genes in the family. And they include the estrogen receptor, the testosterone receptor, that's also called the androgen receptor. They include the thyroid hormone receptor, the retinoic acid receptor, the vitamin D3 receptor, and a bunch of different receptors that have tremendous medical significance. You see why they're called nuclear receptors is because they are functioning in the nucleus, because all these nuclear receptors are transcription factors, and their activity is delayed until they have a hormone. So this is a great example of how development occurs on our planet. Some transcription factors have delayed action. They need something to start their job. Estrogen can be affected by diet, nutrients. It can be affected by external environment. So here we're starting to see evidence that DNA that we inherit plays out over time by regulating the transcription factor activity, sometimes in response to the environment. So we actually have the environment influencing the genetic program. And there's more ways that it's delayed like this, but this is just easily the most fascinating and fantastic. And all these nuclear receptors have that same function. They're ready to go and they're waiting. They're transcription vectors that are ready to go and they're waiting, okay? So notice that the estrogen receptor serves either as a repressor or an activator. So people who study the receptor, and this is just kind of the last point here, is we like to call this the enhancer, right? That's what most biologists like to call it. And you can see in this example that somebody who studies the estrogen receptor hates that term, because it's not an enhancer. It's a repressor or an enhancer, depending on the presence of estrogen. So those people call this an ERE, which stands for estrogen response element. And that's a great term, and I just add it not because we're trying to get into the nuts and bolts details of that, but just that ERE is a great term for that because it could be an activator or a repressor. It's not very good to call it an enhancer. People still do, that's fine, whoever, whatever, who cares, right? We know they are excited about enhancers. Enhancers are cool. This is an enhancer, but a better term for it is an estrogen response element. And the response element just comes from, remember that this is that cis regulatory region. And so these are response elements in that regulatory region. Cool. So that's our third drawing, and it shows us a little hint of how there's this delay in the timing of gene expression. Yep? Lydia, do you have a question? Nope.
So if you were to theoretically, not morally, but if you were to give a young child estrogen, they would produce secondary sex characteristics?
Yeah, secondary sex characteristics. We're mammals, so in the case of mammals, we have internal fertilization, right? So we have a penis and a vagina. If you do this to a fish, there are lots of fishes where if you give them estrogen or testosterone, they'll just switch sex. And in fact, they do that naturally. There are plenty of species of fish that start out as juveniles being all the same sex. And it's only when they go through their puberty and mature that we see a difference between the male and the female. They don't have internal fertilization, so some adult fish switch sexes, not as part of their natural biology. And they can do that because they're either just going to release sperm or egg into the environment. They don't have a penis and a womb, they're just going to release it. So they just make sperm or egg, and that's the difference, right? Yep. Yeah. What's cool is a lot of times those fish have different coloration, right? The male might be really fancy color, and behavior. And those switch too, and the fish switch, So hormones are awesome, and again, they show that kind of delay in the genetic program and also some of the flexibility in the genetic program, right? And I just want to take this chair and throw it out the window and yell. But remember, I did that another year and it was a mistake. Yeah, it's just so cool, right? Because you can get down to the nuts and bolts and you can be like, you're working on cars and you're like, we're working on this car, but it doesn't feel like we're getting ready to go to Florida and party in the car. But we are, it's at that nuts and bolts level where just like as soon as we get the tires on this car, we're out of this snowy place and we're going south. Yeah, it's just like that, you just get it going and it's this molecular reductionism. When we start summing up the parts, you're like, yep, yeah, this is all fitting together, okay? So awesome, super awesome, and we drew that out because you should just be drawing it out, just all the time drawing it out. Now, we're gonna stop, and we're gonna have our first concept chat. So we're doing great, we're gonna run out of time, but we're doing great, and we wanna just introduce this. So here's how a concept chat works. You just randomly make a partner. So we have two students sitting down together, and you talk in just 2 minutes, you talk very quickly about what you think are the 3 most important things we talked about today. Then I'm just going to call on a few individuals. It won't be stressful for me to call on you because you work with a partner. So you'll be like, yeah, what did you and your partner talk about? Ready? Go. So you can slide or you can get up, go. Over the semester, you should say hi to everybody in the class. If you have a group of 3, you have to break it down to 2. Oh, look what's happening here. Total chaos. So if there's a group of 3, like see that group of 3 and that group of 3, so one of you should turn around. Right? Yeah, there it is. Perfect. Yep. Yeah, so it's limited to 2 unless there's an uneven number, and there is. So there's 3. Yeah, all right? Go, go, go.
Yeah, this one's got the cutting and the pacing.
And we just roll through this really fast.
Yes, I knew you
And
Yeah, I didn't know you could get into
this is a good time to sign this. Did everybody sign this yet?
So I understood you could come in and you could make it
Yeah, that was new.
and then I was wondering if I would have
So I was wondering if there was you can have an X amount of experience in here. So we were gonna put 30 from this one and the other 30 from this. But I think we bumped that. Bumped that pretty hard. Okay.
So I guess the first one would be understanding there's 2 different trains. Well, because hybrid is
Yes. What was the difference in the machine? This was, they both were murdered. They are.
What's the different names of the transgenes? Like you have the hybrid one and you have the
None. They're
all just called transgenes?
Yep. Yep. So we're actually just going to do this for another minute. So 3 important things you talked about, and we just roll through it. We do it pretty quickly. Okay, so now I just call on some people, and I just keep track of who I called on up here, so we roll through the thing, and I just call on 3 people each time. So I say, Mallory, what's one thing your group talked about?
We talked about transgenes and also from this
Mm-hmm. Yep. Yeah. So with the muscle video, it's a nice intro because it goes ding, ding, ding. And then actually there could be a delay. And the delay could be for years. It needs something else. Something else has to happen. So that's one reason we talk about the transcription factors and we're making the list, because the list are examples of how the transcription factors get turned on and off. So we've already seen FOXO gets turned off by phosphorylation and sequestering it in the cytoplasm. Estrogen receptor gets turned on. So see, we're making this list because that's what creates these delays. And Juliet was in the same group, but I can call in the same group and say, what's another thing that your group talked about?
With the estrogen receptor, we talked about how it's kind of like the same process as we went over with the muscle, but instead of like it activating, it serves as a repressor and it needs a different.
Yeah, that's just a fancy detail of most of the nuclear receptors is they can activate or repress. You know, that protein must be fascinating. And we'll learn about it in chapter 7 because the way it switches genes off is the opposite of how it switches genes on. And it's another fascinating thing where you're like, oh my gosh, right? Like I often talk about in my classes about you just go get a tattoo of the estrogen receptor. It doesn't matter, you know, male, female, it just doesn't matter because we're all in the same boat with the estrogen receptor and the testosterone receptor. It's doing stuff and it's super cool. Like you could get a full sleeve just going, look at me. Yes, Bree, what's another thing that your group talked about?
We talked about how, like, the difference in the trans genes, like, or the reporter genes, how, like, in the mouse, it'll show muscle differentiation, whereas with the FOXO and GFP, it's showing, like, the location of the FOXO protein and the presence of a growth factor or absence.
Yep. Yeah, so the general term is a transgene. We're going to make a transgene and we're modifying, we're taking DNA in a test tube and we're manipulating any way we want. We can build any transgene we want. Could be one species, could be two, could be three, could be 4 genes like that. On Thursday, Sam's going to talk about putting the coding region from 4 different human genes all together in one to make a super protein. And if you say, I made a super protein, you probably think we must glue the proteins together. No, we just make a gene that has a coding region from 4 different genes, and that's naturally going to make a 4-member protein hybrid gene like that. Yeah, super cool. So transgene is a general term. This reporter term is a cute term because it's reporting. It's just a cute term. Like it's an official term, but it's mostly cute. Because you think of what is it reporting, and it's reporting. You know, it's based on its design, what it's reporting. Yeah, cool. So here's why we do concept chat. The way you learn is by recall. So like we're recalling, that's helping. The more you recall, the better. You're singing a song, right? You got together and you sang a little song, right? Here's another thing that we're doing is you also refer to your notes. So you're taking notes, right? I am remembering something and I'm using my notes as a crutch to help me just remember. So you want to do that. And then you also get to see your neighbor's notes. Some students are really good at taking notes. Not because they're neat necks, but just because they got it organized. I don't know why that's doing that, but we're good. So yeah, now we're going to be done. I have to hand back the quizzes, and when they do, you can take off. So anyway, we got to stop for today. And Thursday, we'll start with Sam talking briefly about school stuff as our first visit from that.